Welcome back, everyone. So, we are going to finish our discussion of protein transport and translocation by talking about our coated vesicles, talking about endosomes, and talking about lysosomes. So, most vesicles that are involved in transporting either those proteins or the lipids for those membranes are going to be transported in coated vesicles. And the reason that they're called coated vesicles is because those vesicles, outside of their membrane bilayer, they have a coat of protein. That coat of protein is on their cytosolic surface, the exterior side facing the cytoplasm. Coated vesicles are involved in transporting those vesicles throughout different parts of the endomembrane system. As you can see in this table here, they start various places, various destinations. They're also involved in exocytosis and endocytosis. And there are a lot of several different types of coat proteins. One coated vesicle has a clathrin-coated vesicle, which means that its coat proteins would include clathrin, AP1, and ARF. They start in the trans-Golgi network and then make their way towards endosomes. Clathrin-coated vesicles that start at the plasma membrane would have clathrin and AP2 as their coat proteins, but still they're going to have the destination of endosomes. COP1 vesicles are going to have COP1 and ARF as their coat proteins. These start at the Golgi apparatus, and then they either go to the ER or the Golgi. COP2 coated vesicles have COP2 and a few other proteins, which begin in the ER, and those vesicles will transport towards the Golgi apparatus. And then caveolin coated vesicles, which are coated with caveolin, they begin at the plasma membrane and probably end up at the ER. When an adaptor protein complex binds to the plasma membrane, or when there is a high concentration of receptors, or when there is a receptor bound to its ligand complexes, that's all going to trigger the beginning of creating the clathrin coat, forming the vesicles, fusing from the plasma membrane towards somewhere else. All of this, binding the adaptor protein to the plasma membrane, having these receptors, receptor ligand complexes, are going to require energy. So here we have ATP, the energy source we're pretty familiar with, adenosine triphosphate, GTP is also another energy source, similar to ATP, but instead of adenosine, it has guanine. So it's still 3 phosphates. You can still cleave that third phosphate and release energy. Assembling the clathrin coat will provide some of that driving force to form the vesicles. And those clathrin units, which this one red area here shows one clathrin unit, you can see it kind of forms into the shape of like a soccer ball with hexagonal sides and pentagon sides, which allows that coat, that clathrin coat, to curve around that budding vesicle. In an actual micrograph, it would look something like this. Those clathrin lattices are lit up in this picture. As clathrin is accumulating around that vesicle, something called dynamin is going to be required. Dynamin is what actually constricts and closes off that vesicle so it can pinch off from the membrane. Dynamin is a cytosolic GTPase. That means that it is going to take GTP and hydrolyze it to release that energy. That energy that comes from releasing that third phosphate is going to allow the dynamin rings to tighten and pull that vesicle away from the membrane. Then, now that we've made our vesicle, we've pulled it away, dynamin has cleaved it off, we're then going to do what's called uncoating, which means removing that clathrin coat from that vesicle. So once we form the vesicle, pretty quickly you can see it's going to start uncoating. The clathrin is dissociating. And this is also going to take energy in the form of ATP. So the uncoating ATPase uses the ATP to take off the clathrin. Then now that we don't have clathrin, there still could be other proteins there, and those other proteins will be responsible to make sure that the vesicle will be delivered to the correct location. To sort and target the vesicles to their proper location involves two different families of snare proteins. SNARE stands for SNAP receptors. There are V snares, the V stands for vesicle, and T snares, the T stands for target. So V snares or vesicle SNAP receptors are found on vesicles, and T snares or target SNAP receptors are found on target membranes. So for example, here is the vesicle with the V snare, the vesicle snare. Here's your membrane with the T snare. Those are going to be able to interact so that that vesicle fuses with this membrane. There's also some tethering complexes that are going to be involved in helping support that complex. So the V snares and the T snares here allow recognition between the vesicle and the target membrane. That these tethering proteins also recognize the vesicle, and they'll hold that vesicle in place so that the V snares and T snares can interact. And then when those V snares and T snares interact, that's what actually allows this membrane to fuse. The membrane of the vesicle and the membrane of the plasma membrane fuse together to create one membrane. This does involve a Rab GTPase. It does involve burning a GTP. And when you have the fusion, all of these proteins, the snare complex, is all going to be released. So when the vesicle gets to a specific destination, the Rab GTPases, and there will be different Rab GTPases for different destinations, they will help lock the snare proteins together, allowing the fusion and using GTP as the energy. After the vesicles fuse, the snares are released from the donor membrane and the target membrane. Okay, so then let's move on to lysosomes and how we are breaking down proteins that are no longer needed. The lysosome is an organelle in the endomembrane system. It is created from vesicles releasing from the trans-Golgi network, accumulating lysosomal enzymes, lowering its pH, and then it'll be a functional lysosome. So it has digestive enzymes that can digest all kinds of macromolecules in biological systems. So all of your four classes, your lipids, your carbohydrates, your nucleic acids, and your proteins. They also have acid phosphatases and other hydrolytic enzymes that will hydrolyze these macromolecules into smaller pieces. And the luminal side, or the inner side of these lysosomes, is going to be coated with glycoproteins. Those glycoproteins give it kind of a slimy, mucousy consistency inside to protect that lysosomal membrane from being degraded itself. Lysosomes are very acidic. They have a pH around 4 or even 5. So they have to be constantly pumping protons into them to maintain that acidity. There are ATP-dependent proton pumps. That means that these proton pumps will pump in protons through active transport, hydrolyzing ATP in the process. And then many of the enzymes inside of the lysosomes are acid hydrolases that will function best at that acidic pH. The lysosomal enzymes can be synthesized by ribosomes on the rough ER as a membrane of the endomembrane system. They are going to be made within the rough endoplasmic reticulum, and then they can be translocated inside the rough ER. Then they'll travel through from the ER through the cis-Golgi network, trans-Golgi network, and then when they get to the trans-Golgi network, they will be delivered in transport vesicles to endosomes. Early endosomes will mature into late endosomes, and at this stage, all of those lysosomal enzymes are inactive because it still has a rather neutral pH. It's not become acidic yet. The pH of that early endosome lumen drops from about 6 to about 5.5, and the endosome is going to lose its ability to fuse with other endocytic vesicles, and at that point, it is going to be fated to become a lysosome. The late endosome will be packaged with stuff to be digested and with newly synthesized digestive enzymes. By packing those digestive enzymes into the endosome, they are not roaming through the cell, digesting anything in the cell. They are only able to digest things that are sent to that lysosome, which protects the cell. You don't have hydrolytic enzymes wreaking havoc throughout your cell. Then finally, to activate a lysosome, you have to activate its acid hydrolases. So to activate the acid hydrolases, you need to drop the pH. So as the pH of that lysosome becomes more acidic, those acid hydrolases will become active, and then your lysosome is able to start digesting those worn out proteins and other materials that are delivered to it. So as that late endosome begins pumping protons in, it functionally matures into a lysosome, or it could fuse with an existing lysosome to have access to that acidity. Sometimes we have cellular structures, not just proteins and molecules that need to be digested, but an entire structure, a mitochondria or something that needs to be broken down. It might be because it is damaged, or it might be because you just don't need it anymore. And the way that we break those down is called autophagy. So auto meaning like self, phagy meaning eating, so self-eating. Macrophagy would be eating an organelle, breaking down an organelle. That organelle will be wrapped in a double membrane from the ER, and that forms an autophagic vacuole or an autophagic vacuole or an autophagosome. So here you can see, here is a peroxisome, here is a mitochondria. You can see it's kind of broken down. It doesn't have its great mitochondrial shape anymore. So you have this autophagic vesicle, this double membrane. See, it's got two membranes to it, coming around these worn out organelles, and it'll be able to break those down. In a microphagy, there's a smaller vacuole that's formed that will be surrounded by a single membrane bilayer, and then this allows these molecules to fuse with these vesicles to fuse with a lysosome and start breaking down those components. There also could be a link between autophagy and cancer. If your cell is unable to break down things that it doesn't need, it would start picking up unnecessary or even damaged pieces in it, and that would start affecting its function, and could turn it possibly into a cancerous cell. Okay, that's the end of chapter 12. This exam, that is your next exam, will have this unit, all of our protein translocation, and all of our discussion of membranes from chapter 7 and 8. If you have any questions, let me know, and I look forward to seeing you in class. Bye.